Okay. So I'd like to start by thanking Stephanie and Melina for inviting me to share this historic moment with you for the very first FAST LATAM conference. So this is a very special moment for the history of this disease in this country. And it's a great honor for me to be able to be a speaker on this occasion. So today, what I want to talk to you about, I see them there, yes, is about what basic research is about with regard to Angelman syndrome. So I am a kind of basic researcher, and I want to tell you about what we do in the lab. Okay, so these are some actual real basic researchers from my laboratory, and you see they're working in the lab, but what are they doing? So the research scientists kind of fall into two different categories. One I would say is basic science, trying to understand the disease, and the other is kind of translational science, trying to make therapies. And so on the very basic research side, we might be looking at things like what are the genes that are involved, as we heard earlier today. We would say, what are the symptoms of the disease? Do some people have more of this symptom and others have more of this symptom? Are some people able to walk? Some people are not able to walk. Why would that be? So that's kind of a basic study, and things like the natural history. What is the natural progression of Angelman syndrome? So these are all kinds of questions that we might ask on the basic science side. Then on the translational science side, trying to make therapies, we ask similar questions. We know what the genes are, so what can we do to modify these genes? What can we do to fix those genes or change the genetic situation? If we know something about the symptoms, how can we improve these symptoms? How do we know that a therapy that we make is going to be useful in a person, right? And then how can we bring this therapy into a person? In other words, not just change the expression of this UBE3A gene in a culture dish, but actually do it in a person. So we need to think about how we're going to deliver that into the body as well. So those are the kinds of questions that we ask in research. So our lab works on trying to develop molecular therapies for Angelman syndrome. So what is a molecular therapy? So as we've heard this morning, Angelman syndrome is a genetic disease, has epigenetic components. These are all have to do with the molecules in the cell. And so some of the more important molecules in the cell are things like the DNA, the RNA, and the protein. And so we're probably all familiar with the fact that you have this DNA that makes these genes, and then that gets turned into RNA, and then that makes a protein. And the protein is the thing that kind of does the work. So small molecule drugs, or we might say medicines, traditional medicines, usually work on the level of proteins. But because this is a genetic disease, we would like to be able to work on this other level, working on the RNA and on the DNA itself. And so that's really what molecular therapy is talking about. Where the problem is on a molecular level, and we want to have a fix, an improvement that's on this molecular level. Now, this would not even have been possible 10 years ago, or maybe even a few years ago, but today we have tools and methods that can actually work on these kinds of molecular levels on the DNA and the RNA. How can we bring this into people? How can we take some kind of molecular therapy and deliver it into people? So today, the most common way that this is being done is with a type of virus called an adeno-associated virus, or an AAV. This is a naturally occurring virus, but the one that we use has been completely genetically changed so that it's not an infectious virus. It doesn't cause disease now. Instead, we pack it and we put inside our therapeutic gene, and we're using this AAV virus as a delivery vector. We call it a delivery vector, so that's going to bring it into the cell. In the past, when we used something like this delivery vector, and we had to bring it into the brain has a blood-brain barrier around it, so it's hard to bring things into the brain. We would have to inject directly into the brain, and you can see from those little black dots in there that it didn't get very wide in the brain. But a real breakthrough happened a few years ago where people were able to redesign this AAV viral vector. And now you can see that it gets very well into the brain. So that's been a real big development lately. The first generation only worked in mice, but nowadays there are actually more than one of these kinds of viruses that can get very good penetration into the brain. So what you're seeing there is a slice of a brain, and what I'm saying is that these vectors can take it in and get all over the brain, and that's what we want. We want something that is not just affecting a little part. We want to hit as many cells as we want, so we can meet or exceed Alison Behrend's 5%. Yes. So at UC Davis, we work with a team of labs, and I'm not going to go into a lot of detail on this slide, but just to say that part of the people that have to do this work are what I might call therapy makers. And that's the group that says, well, if the UBE3A gene is not expressed, what can we do to make it expressed? And we come up with some kind of therapy that we think will be helpful for that. And then asking the question, how do we know that this is going to be useful in a person? Well, we can't test it right away in people. So when we're treating people, that's clinical studies. We work in the preclinical space, so we don't treat people, but we can use something like a mouse model. And mice are good at representing some parts of the disease, not very good at other parts of the disease. No mouse model is perfect. It's not a perfect representation of what happens in a person. And also, it doesn't always answer every question that we want it to answer, but it's the best thing that we have to try these therapies on to know if they're going to work in a person, at least in the beginning. So we work with a mouse neuroscientist who's trained in all of these kinds of special assays, like that rotarod and that other kinds of things that you saw in the movies before. So they can say, when we have this therapy and we put it into a mouse, an Angelman mouse, does it make the Angelman mouse better? That gives us hope that this might work in a person. We also have another group that we work with that looks at the neurons, and they can look at the human neurons, and we could treat those human neurons and ask if our treatment makes those human neurons more like typical neurons instead of like Angelman neurons. So those are the kinds of things that we do in basic research, trying to make the therapy and then test the therapies to see if we think that they're going to be better. And if we're successful in that and we can bring enough information that convinces a regulatory agency like the US Food and Drug Administration that we can start a clinical trial, then we can make that big transition over and actually try it in people. But before we try anything in people, the two things that it has to be is safe and effective. So safe and effective. And so, another message I want to deliver here is that we don't do anything in people until we know that it's safe. All right. So this molecular therapy, is this like something that's really crazy out there, not something that's going to happen for you, but something that happens on TV? No. So this is a list, and it's small writing because there's so many examples, but this is a list of FDA-approved gene and cell therapies. And as you can see, there's a lot of things on that list. And so I'm very happy to stand before you today and say that gene and cell therapy is a thing. This is a thing. This is not like, hopefully in the future, these therapies will be available. You know, we live now in a time when gene therapies are a real thing that are approved, that, you know, a patient can go and have their doctor prescribe this for their patient. So we want to make one of these things for Angelman syndrome. I just want to draw your attention to that last box that's in red. That's Casgevy. Casgevy is the first gene editing therapy approved by the FDA. It was approved only in December of last year, so very new. But the point is, these are the kinds of therapies that we're trying to make to work on Angelman syndrome, and this is a thing, okay? So one thing I hope you take away from what I'm telling you today is that I think there's a good chance of hope, because this is not, you know, abstract talking. These are things that can happen, that are happening. With regard to Angelman syndrome, as has been mentioned earlier today, we know that there's these two chromosome 15 copies, and that usually in the case of Angelman, the maternal copy of UBE3A is either mutated or it's deleted or in some other way inactivated. And so all we have is this paternal copy, the copy that came from my father. That's what we have to work with. However, with all of the strange genetics that happens at this region that make it very complicated, there are some things that actually work to our advantage. So one big advantage is there's a perfectly good copy of UBE3A on the paternal chromosome. It's just off. And so if we could turn that on, that would be a great therapy. And it's a great therapy for a couple of reasons. One is that this is going to give the natural control over this gene expression. So if it needs more of this UB3A protein, it can happen naturally. If it needs less of it, it can happen naturally. So we're not, you know, turning this gene on in some artificial way that you might have too much of it or you might not have the right level. We're just kind of, as they say, turning off the brakes and allowing this gene to be expressed at a natural level. That's not going to be true for lots of gene mutations. So this is a special case for Angelman syndrome where it's actually an advantage. And so you will see that many of the therapies, the molecular therapies, have as their goal to turn on this extra copy. The other thing that I would say is that by turning on this extra copy or in some way reintroducing this UBE3A, we should be able to address all of the types of mutations. So you've seen this slide earlier, and it shows the distribution of the different types of mutations that we can see. So for the largest class, where there's the big deletion, there's no more copy of the UB3A from the mother, but we could turn on the father's copy, and that will give us UBE3A again in the brain. Also with the point mutations, the mutated copies, there's no maternal copy, so we could turn on UB3A again in the brain. And for the other two cases, UPD and imprinting disorders, same kind of thing. We still have a copy of the paternal gene. And so we can use the same kind of therapy to turn it on. Maybe it needs to be tweaked a little bit, but this kind of therapy should work for all types of mutations. So I'm going to go through some of this quickly, and what I want you to take from this is that there are many, many ways that this is being worked on right now. I think Alison Berend talked about many of these, but I like pictures. So we have a case where there's an AAV that's just delivering this UB3A gene, and that's the gene therapy approach. So that's in development. We have another one where we get the cells that are in the body to express this UBE3A protein. And so that's another approach that's approaching clinical development. Everybody knows about the ASO and the wonderful things that that's doing, and we're going to hear more about that in the next talk. But the way that the ASO acts is to allow that UBE3A gene, that red gene at the end, to be turned on, and understanding how that works has led to a flood of other kinds of approaches to do something very similar, either with some kind of CRISPR nuclease or microRNA or some other kind of molecular tool that, really we didn't even have 5 years ago, but now we have these tools and they are being applied to turn on this dad's copy of UBE3A in the brain of mice and eventually in the brains of people. So that's the kind of basic research that is going on for Angelman syndrome. I was also asked to talk a little bit about the role of FAST in promoting this research. So of course you've heard all of this from Alison Berendt just a moment ago, but I'm not a member of FAST, right? So I wanted to say it from my perspective, how FAST has impacted all of this. So certainly they've funded a lot of research of the type that I just talked about. So they've funded a lot of this research and made it possible for many people around the world to apply their innovations to making these kinds of molecular therapies for the treatment of Angelman syndrome. What people may not know is when they got started, one of the innovative things that FAST did was to put together a team of scientists with different types of expertise to try to work on trying to develop these therapies for Angelman syndrome. This is very different than most other foundations. Most of the time, they just put out a call and say, well, if you'd like to work on Angelman syndrome, we've got some money. But they didn't want to do that, right? They wanted to focus in on the problem. And so they got a team together that met every week and talked about Angelman syndrome until finally this effort kind of led to that first ASO therapy that was first in clinical trials. So that was a real innovative approach that FAST took, not just to funding the research, but to really focus it to get it done. And then I would just list some of the things that Alison already talked about. They have the gala every year, and now in Latin America, there's this conference. I would say that these conferences are really important because it brings the community together. It's also an opportunity for the scientists to meet and talk with each other. And so hosting these galas and these conferences and the scientific summits, it's all very important. And so it's all perpetuating and increasing the rate of things that are happening for Angelman. We also heard about the A-bomb, which is looking for clinical outcome measures. This is very important to transitioning into this clinical phase. And so this is a super important effort that involves many, many people. It's led by several people, but the top leader is Alison Berendt. So this is another effort that they put together. Other things like trying to train other clinicians how to perform the injections that need to be performed in these kinds of treatments, through the Rush Center, and you're going to hear from Dr. Barry Kravitz in a few minutes. But this is another thing that FAST has been doing to try to make these therapies accessible to everyone. And as you heard also, now they have an accelerator program that can help bring this transition from the basic research into the clinical research, where all of us clinical research or preclinical researchers would like to go. And then the last thing I would say is that there's now also this conference in New York City where FAST is essentially training and collaborating with other foundations so that the knowledge that they've acquired can be shared for other rare diseases. And so, you know, the impact that FAST is having is not just in Angelman syndrome, but, you know, for the broader rare disease community. So it's really been amazing to be part of this, and FAST in every country. So then I want to end with just talking a little bit about the work that goes on in my lab for Angelman syndrome. And so, as I said before, when we think about the antisense oligonucleotide that's in clinical trial, it works by turning off an RNA that is preventing the UB3A from being turned on. And they do it at the very end of that gene with this ASO. But in some early work that we did, we were able to show that a similar kind of approach could be to turn the gene, that long blue RNA off at the point where it's made, so early on. And we're doing that with something called an artificial transcription factor that I'm not going to describe very much, but just to say that we were able to design this molecular therapy to turn off that thing that is stopping UB3A from being turned on the father's chromosome. And so we tested this, and several people in my lab were responsible for these data. And so I think you might have seen this slide already, where we were able to put our artificial transcription factor into an AAV virus. Now you know everything about AAV, right? And so we were able to inject that into a mouse model of Angelman syndrome. We could see in panel D there that there's the wild type, or a natural mouse that doesn't have a mutation in UB3A. It has a lot of brown dots. That's the UB3A protein that we're seeing under the microscope. In the middle lane there is an Angelman mouse, and it has less of those brown dots. And then when we treat it with our therapy, we see more UB3A turned on. That's exactly what we'd like to see. But we didn't just stop there. We looked to see, did this make improvements in the mouse? So we turned to our mouse expert and said, if we inject this therapy into the mouse, does the mouse seem to get better? And so they performed a battery of tests. I'm just showing you a couple here. And these tests have to do with how the mice move around and their coordination and their activity. And all you need to know about this is that black line on the top is the wild type mice, the good mice. The blue line on the bottom is the Angelman mice, and you see that they're different. And our treated mice are the red mice. And all you need to know is that that red line is closer to the black line than it is to the blue line. That's good. That means it's good. So we think that this is a good approach for the treatment of Angelman syndrome, and we're working very hard to bridge this gap now, to make this transition from our preclinical work and in the research side to try to prepare information for the US FDA to transition us to having an in-person clinical trial. So I'll just end by thanking the people in my lab that have done all this work, and thank all of you for allowing me to speak to you today at this very first FAST LATAM convention. Thank you very much.